Hello, good afternoon. I hope everyone has been able to have a look at the posters and more importantly, had some lunch. I'm Fan Chung. I'm a clinician and I'm a professor of respiratory medicine at Imperial College. I'm not part of REMP and I'm very honored to have been invited to chair this session this afternoon. Um, I've got a lot of interest in aerosol because I head the COVAIR EPSRC funded project, which has the main uh, the main aim is to look for um, aerosols of COVID in the air, which is not an easy task. So this afternoon we are going to have a continuation uh, of the um, of the transmission of COVID-19. There's going to be three talks, and um, and I'd like to introduce the first talk to be given by Marc Stettler. So Marc is the leader of the Transport and Environment Lab and also the EPSRC funded Center for Doctoral Training in Aerosol Science, and therefore an expert in aerosol science. And so he will talk to us. He's from Imperial College, and of course, he'll talk to us about, as you can see, the source terms for benchmarking models um, of aerosol and droplet disease transmission. So, Marc.
Thank you for the introduction, and it's a pleasure to speak at this workshop. I'm going to be talking about source terms that we're proposing to use for benchmarking models of aerosol and droplet disease transmission. So this work has come out of RAMP, one of the RAMP groups, subgroup 2 on aerosol transmission, and so is the product of several authors listed here. So if we kind of look back to our simplistic picture of transmission via aerosols and droplets, then we've got a spreader on the left-hand side exhaling particles. They've also got a sort of exhaled flow rate of air leaving their mouth. That is breathed in by a recipient on the right, but in between that we have aerosol transport and evaporation processes. So we in our picture, the larger droplets are falling to the ground more quickly, the smaller droplets are remaining airborne for longer distances, but in that distance as well, there's evaporation of particles of the liquid water from the particles, meaning that those particles are shrinking. And then the recipient is inhaling them, and the size of the particles at the point of inhalation determines the deposition within different parts of the airways. And so several groups around the world and governments are modeling this process. And one of the important inputs to this modeling process is these source terms on the left-hand side. So they include the exhaled airflow, the aerosol concentration and size distribution, the viral load within particles, and also the particle composition, which determines the equilibrium particle diameter after evaporation has occurred. And the modeling effort is really accelerating, and looking back to October 2020, I did a search for exhalation droplet model and 22 results turned up. I did the same thing yesterday and 4,100 results turned up. So, I'm not suggesting that these are the most important papers to read or all of the papers in the literature, but it just gives us a sense of scale of the acceleration of publications coming out on this topic. And the issue is that not all of these models and modeling approaches are using the same set of terms, which means that it's not possible to evaluate whether differences in results from different models are a result of differences in assumptions on the source terms or differences in the representation of physics. So our objective is to propose a set of source terms to be used for benchmarking of models, and these source terms are reasonably well known and can be used by a range of modeling approaches as the basis for model intercomparison. We focus on breathing and speaking initially, at least, because these are the highest frequency events. So of course we recognize that particles are produced from coughing or sneezing, but breathing and speaking are higher frequency, and certainly so for asymptomatic individuals. And we define parameters for exhalation, exhaled particle size distribution and composition, and environmental conditions, and viral load, although we hesitate and hold back from providing a single value that should be used for intercomparison, and instead discuss ranges and important considerations. So if we dive in and look at exhalation, so there's a paper from 2010 from Gupta et al. That looked at breathing and speaking, and they characterized flow rates out of people's mouths and noses during these activities. On the left-hand side, breathing you can see is represented by an almost sinusoidal function, but the period of inhalation and exhalation is different, and so is the amplitude. There's a correlation between the period and amplitude with body surface area, which is also correlated with individuals' weight and height. On the right-hand side, speaking is a lot less regular. Okay, it's very irregular in the flow pattern, and therefore very difficult to characterize in terms of a nice, smooth function. But we can see that the flow rates differ significantly for different letters or numbers that are spoken, and therefore what we propose is that rather than try to capture all of this variation for the purposes of model comparison, it might be appropriate to assume a nominal exhaled flow rate for speaking. So in this paper, they were also able to characterize the jet angles of exhaled flows, so from the mouth and from the nose. So to summarize our source terms for exhalation, breathing can be represented by a piecewise sinusoidal function, where the first part is inhalation and the second part is exhalation. And the figure on the left shows a typical flow pattern for an average male in the UK and an average female in the UK. And you can see that both the period and the amplitude of the sine waves are different for inhalation and exhalation. Some models are able to account for this dynamic exhalation flow, whereas others are more steady state and would require a nominal average flow rate, exhaled flow rate of 10.6 liters per minute, which can be obtained by integrating the exhalation part of this sine function. For speaking, as shown in the previous slides, the flow rates are far more variable, and therefore it would make sense to use a nominal average exhalation flow rate of 12 liters per minute, or 0.2 liters per second. And this is within the range of other measurements from another study from 2016. But note on this study, on this figure on the bottom right-hand side, the large variation between people. There is consistently a larger airflow from males compared to females, but there is significant variability amongst the population. So in terms of the kind of jet flows, we're summarizing them here. So for the nose breathing, they're defined in terms of angles as viewed from the front and from the side, and on the right-hand side here for the angles for mouth, exhalation from the mouth. But caution, all of this data is for healthy individuals, and there's a lack of information on individuals with specific diseases and specific symptoms. Also caution that normally there is an asymmetry in nasal flows due to anatomical, physiological, and disease factors that could shift and alternate the asymmetry over time. So it's a simplistic representation just to split the nasal flow between two nostrils, but for the purposes of model comparison, we think that that's appropriate. So what do we know about exhaled particles? Well, they are they've been measured to be on the range of 0.01 to 1000 micrometers in diameter. However, measurement techniques differ across this size range. So for particles greater than 10 micrometers, common techniques include microscopic analysis of droplet marks on slides, and also optical techniques. And for particles less than 20 micrometers, an instrument that's been commonly used is the aerodynamic particle sizer, which is only sensitive in the range of 0.5 to 20 micrometers. So in many cases, studies don't measure both. They either measure the larger droplets, or they measure the kind of less than 20 micrometer particles. Also, there's significant differences in whether studies account for the full flow of exhaled air, or they just sample flow, and also whether studies dilute or take the flow undiluted. These measurements are also affected by ambient conditions, and particles of diameter less than 10 micrometers will shrink to their equilibrium diameter in the timescale of less than 1 second. So this is affecting the measurements that we see and have been reported. In some cases, studies try and correct for this. In other cases, there is no correction applied, and therefore caution needs to be applied when interpreting the particle size distributions. We know that for speaking, there's significant differences due to loudness and vocalization. And studies suggest that there's significant variability amongst people, so high interperson variability in the so-called super spreader phenomenon. The literature, we think that there's 3 formation mechanisms, so bronchiolar and laryngeal formation mechanisms both lead to a mode in the particle size distribution of between 0.5 and 5 micrometers. And there's an oral mode, which is formed of larger droplets, so greater than 10 micrometers. And in this figure here, what we're doing is comparing different studies on the left-hand side for breathing and on the right-hand side for speaking. So I apologize that the slide on the right-hand side is a little bit busy. The different colors denote different studies. The Johnson et al. Paper, which is highly cited, shown for breathing and for speaking. More recent measurements from Gregson et al. Shown for breathing on the left-hand side, and then on the right-hand side for speaking, shown for both a low volume vocalization and a high volume vocalization. And we can clearly see the effect that higher volume, louder vocalizations lead to larger concentration, so higher magnitude concentration. We have a size distribution from Asadi et al., which most people will be familiar with. And then we have 3 studies which are focusing on the oral mode of particles, Chow et al., Shi et al., and Dugoid from 1946. And so what's immediately obvious is that there's a significant variation amongst these reported particle size distributions, both in terms of the smaller particles related to the bronchiolar and laryngeal modes, and more significantly for the oral mode of particles, where we see orders of magnitude difference in concentration and significant differences in the reported spread of the distributions. I want to highlight this area of the graph here, which shows 2 size distributions reported from Johnson et al. The dashed line is uncorrected, i.e. as was measured at the sampling point, and the solid black line is that particle size distribution corrected for particle shrinkage. So in this study, they applied a factor of 1/2 to correct for the shrinking diameter of particles as a result of evaporation. And we can compare that dashed black line directly to the solid lines of Asadi and the orange lines of Gregson, because they were also uncorrected. And note that they're in relatively good agreement, but that the particle size distribution and the mode of these bronchiolar and laryngeal particles is likely to be larger by a factor of 2 or so at the exit of the mouth. So for the purposes of model comparison, Johnson et al. Is the only study here which provided both measurements of the, provided measurements of all of the three modes, the bronchiolar, laryngeal, and oral modes. And that's the study we therefore use as the basis for model comparison, but we do encourage modelers to use these different particle size distributions and test the sensitivity. So this is just a mathematical representation of that size distribution. It's formed of three modes. So the bronchiolar is mode 1, then we have the laryngeal as mode 2, and the oral as mode 3. And so this graph now shows the size distribution number weighted in the top left-hand corner, the size distribution volume weighted in the top right-hand corner, and then the cumulative number fraction in the bottom left and cumulative volume fraction in the bottom right. And what we're showing here is that the total volume of particles emitted is dominated by, at least in speaking, dominated by the oral mode of particles. And so if we're comparing speaking and breathing, then the number emissions rate, now combining our concentration and the flow rates, exhaled flow rates. For speaking, the number emissions rate for speak is 2.6 times higher than it is for breathing. But if we're then looking at volume, the speaking emissions rate is 5 orders of magnitude higher than breathing. And that's because for speaking, we have this oral mode of particles, whereas for breathing, we don't. And this oral mode of particles are larger and volume goes diameter cubed. Therefore, this oral mode contributes significantly more, much more volume in terms of particle volume emitted than the bronchiolar or laryngeal. So for composition, we are proposing a composition, a model composition used from this paper, so Bejarano and Ma, the 4-component model. So saying that the particles are made up of water with sodium chloride, mucin, a protein, and DPPC, a surfactant. Okay, and we provide the properties of those components, but note that real respiratory fluid is much more complex than this, contains a variety of salts, proteins, and surfactants, and that the total protein content can vary widely from different respiratory fluids and for different individuals. In terms of viral load, the take-home message is to be careful. The evidence is incomplete. It depends on the pathogen, the individual, the stage of infection, and respiratory fluid. We caution against using nasal swab samples because these are often diluted in transport media by a factor of 10 to 100, and we're not sure that that's been explicitly accounted for in studies to date. It is clear that there have been studies which have shown that SARS-CoV-2 is present in exhaled breath condensate, so up to 10 to the -1 gene copies per cubic centimeter of exhaled breath has been reported. However, there is a large variation in this viral load. So typical values that have been reported and used are at 10 to the 6 copies per mil. But we know that there's a variation in that viral load over time, and that it usually peaks around symptom onset. However, it's important also to note that just because we measure RNA copies, that doesn't necessarily mean that there's an infective virus present. And one study has shown that that there was one infective virus determined by viral culture methods for every 1.6 or 1.7 times 10 to the 4 gene copies that were measured per milliliter via PCR. So to summarize, we propose a set of source terms to be used for model benchmarking. We we recommend that results are reported in detail and that all physical and mathematical models are described, including description of the model scenario with a diagram, dimensions, and all boundary conditions, that the results are resolved with respect to particle transport and dynamics as a function of particle size, and that distributions of number and volume concentrations as a function of time and space are given. There is significant variability in all parameters that I've been speaking about, but the statistical distribution in the population is not well known. In many cases, studies are not directly comparable, and therefore we encourage modelers to conduct uncertainty and sensitivity studies. I'll leave this slide up just for a minute. This is our kind of consolidated set of source terms. We do have a paper in preparation that is due to be submitted very soon. But if you need to, if you want to have a look at these source terms for yourself, then please do just get in touch with me. Thanks for listening.
Great. Thank you very much, Mark, for kicking off this afternoon, and you're right on time, and thanks very much for that. I've got one question to kick off here. And the question is about the particle shrinkage and also growth, particularly in high humidity, and others will more great. So it is assumed that particles only shrink, but surely some will grow in high humidity and others will agglomerate. I don't know whether you can address this issue. I mean, if I could also perhaps enlarge on this question. I mean, for particles, aerosols that might contain COVID that we inhale ourselves as the recipient, I guess once it gets into the airway, it probably will grow in size because the airways are fully saturated.
Yeah, so we typically think of our exhaled breath at the point at which we exhale it to be near 100% relative humidity, and then ambient conditions typically are lower than that. And so there would typically be evaporation rather than any further growth. Obviously, that could be different if you're in a highly humid environment or a supersaturated environment. In terms of agglomeration and aggregation, I think the concentrations of particles that we're talking about are very low, so of the order of 1 or 0.1 particles per cubic centimeter, and therefore aggregation effects are likely to be so I would think negligible, at least to first order. And then, as you're right, as you inhale particles again, you're inhaling from a low humidity into a high humidity environment, and that could lead to condensation of water vapor onto the particles in the respiratory tract, and change their size and aerodynamic properties, therefore, and where they deposit.
And therefore, they might deposit really in the central airways, because you'd assume that if an aerosol size of 1.5 micron would grow to a much bigger size. Do we know what sort of size? I mean, if it's a 0.5 micron that gets into the airway, what sort of size will you get to?
Off the top of my head, unfortunately, I don't.
Okay.
But yeah, I'll refrain from guessing.
Thanks very much. I think that's the only. Yeah, maybe I could. But okay. Yeah, sorry. There's another question that came up. Why is the size distribution function? Why the size distribution function is bimodal in slide 11? On slide 10, all distributions mostly log normal. Why is distribution of stationary? Could you answer that quickly?
Yeah. So the size distributions are actually, well, there is a mode related to the bronchiolar, there's a mode related to laryngeal, and there's a mode related to the oral formation. So there's 3 modes specified by Johnson et al. And for papers that have looked at just the particles less than 20 micrometers, they've also reported bimodal distributions related to the bronchiolar and laryngeal formation processes. They that, I mean, the distributions are not perfectly log normal. And actually for the larger aural particles in some of the studies that have used slides or optical techniques, the distribution isn't log normal. And in fact, you could compose some of those, some of the data that I showed were composed of 3 log normal distributions. And so that's, you know, for some respects, that's just a representation of the data. We don't know exactly why the distributions are shaped that way in those cases or for those particular studies. With regards to why these distributions are stationary, I'm afraid I don't quite understand what that means. If that means in terms of time, then the measured distributions are measured over a period of time. So that might be of the order of a few seconds over which the exhaled breath is being sampled and measured. And so what's reported then is, in most cases, to my knowledge, is the average particle size distribution over that period of time where the authors have considered that what they're measuring is actually exhaled particles.
Right. Well, thanks very much, Mark. There's another question. Maybe you could answer that to Peter Fleming, which is a question following your answer to his first question. Thanks very much, Mark.